Histology of muscle. Skeletal muscle tissue. Muscle histology. Elongated cylindrical cells equals muscle fibers. Plasma membrane equals sarcolemma. Transverse, T, tubules tunnel from surface to center of each fiber. Multiple nuclei line the surface of cell. Cytoplasm equals sarcoplasm. Muscle histology. Throughout sarcoplasm is sarcoplasmic reticulum. Stores calcium ions. Sarcoplasm contains myoglobin, red pigmented protein related to hemoglobin that carries oxygen. Along entire length are myofibrils. Myofibrils made of protein filaments. Come in thick and thin filaments. The sarcomere filaments overlap in repeating patterns. Unit structure is called sarcomere. Separated by Z discs. Darker area equals a band associated with thick filaments. Age zone has no thin filaments. I band has thin filaments, no thick filaments. Functional anatomy. Thick filament, myosin, has movable heads, like heads of golf clubs. Thin filaments, actin, are anchored to Z discs. Contain myosin binding sites for myosin head. Also contain tropomyosin and troponin. Tropomyosin blocks myosin binding site when muscle is at rest. Sliding filament mechanism. During contraction, myosin heads bind actin sites. Myosins pull and slide actin molecules, and Z discs, toward age zone. I bands and age zones become more narrow. Sliding generates force and shortens sarcomeres and thus fibers. Neuromuscular interaction. Nerve signal triggers muscle action potential. Delivered by motor neuron. One neuron can trigger one or more fibers at the same time. Neuron plus triggered fibers equals motor unit. Neuromuscular interaction. Neuronal ending to muscle fiber equals neuromuscular junction, NMJ. Synaptic end bulbs, at neuron terminal. Release neurotransmitter. Muscular area equals motor end plate. Between the synaptic left. 1. Axon terminal. Axon collateral of. Somatic motor neuron. Sarcolemma. Myofibril. ACH is released. From synaptic vesicle. ACH binds to ACH. Receptor. Junctional fold. Synaptic vesicle. Containing. Acetylcholine. ACH. Synaptic cleft. Space. Motor. End. Plate. R. Neuromuscular junction. B. Enlarged view of their neuromuscular junction. C. Binding of acetylcholine to ACH receptors in the motor end plate. Synaptic end bulb. Neuromuscular junction, NMJ. Synaptic end bulb. Motor end plate. Nerve impulse. Muscle action. Potential is produced. Na plus 2. 3. Action at NMJ. Release of acetylcholine, ACH. Diffuses across cleft. Activation of ACH receptors. Generation of muscle action potential. Repeats with each neuronal action potential. Breakdown of ACH. Contraction trigger. Muscle action potential CAR2 plus release from sarcoplasmic reticulum, senior. CAR2 plus binds to troponin. Moves tropomyosin off actin sites. Myosin binds and starts cycle. Contraction cycle. Myosin binds to actin and releases phosphate group, forming cross bridges. Cross bridge swivels releasing ADP and shortening sarcomere, power stroke. ATP binds to myosin, release of myosin from actin. ATP broken down to ADP and Pi activates myosin head to bind and start again. Repeats as long as Ca2 plus concentration is high. Relaxation breakdown of ACH to stop muscle action potentials. Ca2 plus ions transported back into senior lowering concentration. This takes ATP. Dopamine covers actin binding sites. ACH diffuses across synaptic cleft, binds to its receptors in their motor end plate, and triggers a muscle action potential, AP. Nerve impulse arrives at axon terminal of motor neuron and triggers release of acetylcholine, ACH. Synaptic vesicle filled with ACH. ACH receptor. Acetylcholinesterase in synaptic cleft destroys ACH, so another muscle action potential does not arise unless more ACH is released from motor neuron. Ca2 plus muscle action potential nerve impulse senior contraction power strokes use ATP myosin heads bind to actin, swivel, and release. Thin filaments are pulled toward center of sarcomere. Troponin tropomyosin complex slides back into position where it blocks the myosin binding sites on actin. Muscle relaxes. Ca2 plus active transport pumps. Ca2 plus release channels in senior close and Ca2 plus active transport pumps use ATP to restore low level of Ca2 plus in cytoplasm. Ca2 plus binds to troponin on the thin filament, exposing the binding sites for myosin. Muscle AP traveling along transverse tubule opens Ca2 plus release channels in their cytoplasmic reticulum, senior membrane, which allows calcium ions to flood into the cytoplasm. Elevated Ca2 plus 1, 2, 3, 4, 9, 5, 6, 7, 8. Transverse tubule, muscle tone. Even at rest some motor neuron activity occurs equals muscle tone. Keeps muscle in a state of readiness. If nerves are cut, fiber becomes flaccid, very limp. Production of ATP for muscle contraction. Aerobic cellular respiration. Production of ATP in mitochondria. Requires oxygen and carbon substrate. Produces CO2 and H2O and heat. Fatigue, inability to contract forcefully after prolonged activity. Limiting factors can include Ca2 plus, creatine phosphate, oxygen, buildup of acid, neuronal failure, oxygen use after exercise. Convert lactic acid back to glucose in liver. Resynthesize creatine phosphate and ATP. Replace oxygen removed from myoglobin.